Martin Jechlinger, from EMBL Heidelberg, speaks about exploring treatment-resistant breast cancer.
And why did we want to kind of like study breast cancer? Because as we all know, there were spectacular successes actually recently in the last decades in treating primary breast cancer patients. So we have survival rates, initial survival rates, I should say, of more than 90%. And the initial I'm stressing for the simple reason that, in fact, the main problem in breast cancer and the reason for most breast cancer-related deaths is the progression to recurrent disease. And these recurrent tumors are kind of like hard to treat. They are almost incurable because they are deaf to the initial successful treatment. So for that reason, there's a huge interest in trying to understand which cells survive therapeutic intervention that was successful at the beginning and survive as residual disease, and how do they kind of like progress to relapse. And that's a hard question to ask because, as probably the clinicians can explain better, we only get patient material from these stages, from carcinoma stage and from recurrent tumor, but in between, that's basically a black box. So you don't get the tissue. You can't really study that residual disease. And for that reason, we, as more basic scientists, decided to kind of like try to address these areas where we don't understand the progression of the disease with model systems. So we are using mouse model systems, and these mouse model systems are regulatable. So these are second generation mouse model systems where you can induce the oncogenes like MYC and HER2 that are really important in adult breast cancer in human. You can induce that with doxycycline and the inducer, a dimer, to be expressed in the adult mammary gland specifically. So you turn it on, you get, here in the histology, you see the initial stages of a growing tumor. These grow quite big, but despite being about 10% of the body weight of the mouse, you can deregulate that. So you basically take away the inducer, you turn off the oncogene, you mimic the perfect drug treatment if you want, and you get residual disease. Or you get a normal-looking mammary gland, as you would say. And these mice are having a functional mammary gland that can nurse their pups. And mimicking kind of like the problem that we see in the clinic, 90% of these mice come back with a tumor, with a recurrent tumor after 6 months. 6 months is a long time in a mouse life. So we basically mimic the situation, but we still have the same problem. We don't know which of these cells are actually causing the relapse at the end of the day. This is a stochastic relapse, right? And for that reason, we resorted to basically a technical trick. We went into organoid technology and generated out of that mammary gland of the mouse a primary organoid in a 3D matrix where we can recapitulate the whole process if you want. So we can induce the oncogenes, we get growth, we get this tumor correlates, and when we turn off the oncogenes, we get similar looking kind of like sacs, acini, that we could prove then are the source of relapse. We transplanted these cells into the cleared fat pad of mice and got similar looking relapses after some months. We also did serial transplantation, if you want, into different gels, kind of like transporting them, and also got, as you can see here, stochastic outgrowth. So we are pretty sure that we now have a pure population of residual cells that can basically give rise to relapse that we can study now because we can see it. We can basically take them out of the dish. So we did stainings, right? I mean, so you can see they are, again, repolarized. The tight junction markers align the lumen, the basal marker, like alpha-6 integrin, are the outside where they should be. The GM130 protein, Golgi protein, is at the right place, looks very normal. Tumor, a vast mess with, we have a human transgene for MYC. So this human transgene is stained for here, and it's completely off again, so there's no leakiness of the oncogene. They look normal in all respects, and still, these ones give rise to relapse, whereas these don't. What is the difference? And of course, we set out then to transcriptomic profiling. There's no surprise that in this PCA analysis, the overall difference is that the normal cells are vastly different from the tumor cells. The real question was like, where do these residual cells reside? Are they back? Because you could, assume so, to the normal phenotype. No, they are completely different. So there are vast differences, and in these differences, we suspect it must be the difference also on the property or on the possibility that they give form to relapse. One of the first kind of like bigger targets that we could identify was deregulated lipid metabolism. Very much to our surprise, we saw all these proteins upregulated, the whole pathway upregulated. And more importantly then, due to the overactive import and also into mitochondria and degradation over beta fatty oxidation, you get as a result hyperactivity or high levels of reactive oxygen species, which again work on DNA damage and so on and so forth. I'm not going to go into detail, but this is kind of like the metabolic hallmark that we found that we could verify. And once you know what you're looking for, you can then go into the mouse again and see that the relatively normal-looking regressed mammary glands, so after the tumors shrink again, do carry those features, these proteins upregulated, waiting for, or basically priming the cells for possible relapse later on. Importantly, when you know what you're looking for, now can go back to new adjuvant treated patients, right? And reanalyze these targets or these proteins that you have identified in the model system for their overexpression or their existence in post-therapy tissue. Okay, sorry. And, okay. Did you hear me before? Okay. This is in a HER2 positive breast cancer. So that kind of like mimics our model, but very much to our surprise and also, putting this residual disease in a different perspective, we also saw, for instance, FAS-N and other proteins that we identified as targets in the luminal breast cancer or in triple-negative breast cancer in the residual disease upregulated. And we could then also, in retrospect, kind of like identify and verify quite a few of our pathways. And encouraged by this, we decided to go deeper, right? So we basically went then into very, let's say, involved experiments in 3D cultures where we did lipidomics, metabolomics, kind of like in a very fast way to kind of like get the metabolites out, overlaid it with the transcriptomic profiles, and did a multiomic, also guided by flux analysis, picture of the cells, basically highlighting the difference of the residual cells versus the normal cells, and basically trying to understand what would be synthetic lethal targets, as was already explained by Professor Zuberti-Fugger, to the initial treatment. Yeah, what else could we do to the residual cells that wouldn't touch the normal cells? And we found a few on top of the overactive lipid pathways and lipid synthesis, we found the whole glycolysis pathway upregulated in respect to normal cells. So you have in blue glucose downregulated because it's imported in the downregulated in the outside, and the whole glycolysis upregulated. But then you see something like a Warburg effect. Instead of the majority of it getting into the TCA cycle, you get lactate, and lactate exported. Probably also hinting on other targets like, for instance, monocarboxylate transporters in order to stop that process, making cells vulnerable on that end. You have the TCA cycle itself dysregulated. You have all these anabolic processes for amino acids and fatty acids upregulated still, as if they were still a tumor cell, right? So they behave kind of like in a way for their metabolism as if they would still be in this highly proliferative tumorigenic state while they are resting and polarized. You also have the urea cycle upregulated and old tumor markers, well-known old tumor markers secreted in these cells like ornithine, putrescine. So that was quite interesting, but as often also overwhelming, right? So you need then to narrow down and find a test system where you can actually go through those targets. What we decided to do is designing a 96 well-plate assay that is suited for 3D cultures, right? So we have 96 small 3D culture in this plate that we need to kind of like control. So we needed to kind of like measure the growth and calculate the cell mass during the growth phase, so to compare well to well. And then when you basically put in your inhibitor, your drug that you want to test, you basically get the dissolving or reduction in cell mass, and you can also multiplex this with just normal viability and death assays on a plate reader. So that way you basically have 5 curves that should meet at the IC50. And you can test, you know, the strategy that is basically right now prescribed. You can do synthetic lethal targets to it. You can do mono treatments. And one example I just wanted to tell you or show you that came out of the fact that we found a few of those enzymes in glycolysis upregulated, like hexokinase 2, specifically in cancer cell and residual disease, but not in the normal cell. So we kind of like tried the inhibitor to that. And here you see the results. So you see that specifically the residual cells are vulnerable and undergo cell death, whereas the normal cells are untouched. You might also see that the tumor cells don't really react to that single treatment, but they still have a lot of other pathways. So that might rescue kind of like just the treatment of the 3BP. But that gives a handle to, you know, as we were discussing, synthetic lethal treatment. So you treat with the HER2 inhibitor and you hit the cell on the other hand on the glycolysis pathway, and you might just be reducing the substrate for relapse by that way. So that's kind of like what we were heading for, and I mentioned this, or you might have thought it. I mean, you know, they are very similar in these respects to the tumor cells. So we did basically Metabolics. We tried to see like how do they actually look like, you know, for open transcribed regions, and in fact, they are very close. The tumor cells and the residual cells are very close and different to normal cells. And when you look at the methylation proportions, right, normal cells, they look normal. So you have a lot of housekeeping genes, open methyl regions, and a lot of, you know, methylated regions where it's blocked. Tumor cells, it's well known. You kind of like loosen up this. You have leakiness. You have a lot of targets being transcribed. And yes, this was the question. Residual cells do keep this, and that's why we basically say that based on our experiments and on what we could verify then in model systems in the mice, on mice tissues and on patient tissues that we got back at that stage, we kind of like see these residual cells as a specific kind of like in a specific state that makes them vulnerable. And that's the good news actually, right? So you have them despite looking normal, not behaving, not being proliferative, in our hands in the breast case, different from normal cells. And that gives us targets. We termed this oncogenic memory, and we are basically now trying to, with our assays, to kind of like go through the different targets and then go back to the model systems, go back to the patients in order to see if we can help them. What I told you until now is actually very nice. It just has one of these problems where we thought we can actually improve the situation, because one of the facts there is that also in the best transgenic mouse model, you transform a whole tissue. It's not a single cell that gets tumorigenic, it's not a cluster of cells as you would normally see that. And with the system that we had in hand, we thought that we could actually get better at that, because what we wanted to do is also, since we have this now in a dish, with light sheet microscopy, to film the process and to see the emergence of single clones. So this would be the system that I presented to you before, right? So you have 3 transgenes. We just basically mark a few cells with a nuclear marker, so H2B bound to GFB, and then when you induce, all of them kind of like proliferate, and you have the green cells interspersed. They're just green, so we can track them better. But what we're thinking of doing is just basically take the 2 oncogenes and put the inducer together with the green protein into the viral vector. So now, in theory, only the cells that are marked are having the propensity or the capacity to kind of like induce the oncogenes. And that should look like that. So only those would grow a tumor within an otherwise healthy surrounding. And this is kind of like, so this is cutting a very long story short, as you might imagine. This is a film over 3 days. With the light sheet microscope, you're that little phototoxic that you can really see that exactly what we were hoping for would kind of like happen. So you have these clones, these green clones growing out, and you see that the other cells are kind of like, I mean, they're wobbling around, but this is normal tissue. They don't do too much. So this would have been perfect, and we would have wanted to proceed to de-induce and kind of like see how they reintegrate and how this residual disease survives and where we could interfere. That was the aim, right? Now, 40% of the situations were like this. Nothing happens. So these cells would just stay stable, and we thought initially, well, probably they don't express the oncogenes, probably something technically went wrong. We could exclude that. And as you can see here, with the antibody against MYC, that is basically for the transgene, we saw that in fact they express the oncogenes. So they are held back by the surrounding. Interesting. So why? Okay, this is a technical slide. I think I just skipped that. This is just showing you that this was a painful process to do single cell tracking over 3 days, but we managed. And what we found out is that there were different clones, right? I mean, within the same organoid, you even had some cells that would kind of like proliferate and grow as a tumor, whereas other ones here in blue, the blue marked ones, they would just stay the whole time and do nothing. So we basically termed this proliferative phenotype, non-proliferative phenotype, right? And that kind of like on itself was an interesting observation, because that way you basically, what we switched now our question to was like, how can it be that you express an oncogene and you just stay silent over 3 days, right? What holds you back? And we basically tried to also then, with a lot of data, go through these situations and wrote down all the different scenarios that came to mind. And what we ended up with, that it's actually, in mathematical model, the decipher element is that you have minimum 3 cells in a smaller volume within the organoid that basically interact, and therefore they can grow out a tumor. That raises a lot of questions. How do they basically, what do they need to do? Do they need to kind of like interact in a way that they kind of like wobble the epithelial environment and break up the epithelial barriers so they can break through? Is it a very short range paracrine signal that helps them do that? And all of these questions we're addressing right now. So what we are implementing into our workflow of the microscope is then also a caged marking approach of different regions, where we can basically, like in Star Wars, highlight some cells of interest, some regions of interest, get them out, and do transcriptome analysis and you know, multiomics actually, and try to see what's going on in these areas to explain the phenotypes. Yeah, so this is how we basically function. Wanted to give you an overview, and thank you for your attention. We have a lot of questions still left in our lab, so I'm waiting for yours, and I thank the people that did the work, former and present members in the lab, all the core facilities and collaborative labs that help us do this very interactive and very interdisciplinary research, and of course the funding organizations. Thank you very much.